All right, so this is our last video of this semester, and so this video is going to cover adaptive immunity. So we're going to talk about specific immunity. Last time we talked about innate immunity, things like inflammation and generalized responses, and so this we're going to talk about adaptive immunity. So adaptive immunity is going to be a specific response. So you're going to have pathogen exposure for this immunity to take place. So this immunity kicks in after you've been exposed to the pathogen, and then you're going to start creating specific cells that are going to be able to respond. They're going to respond specifically to that particular pathogen. So it has to occur after pathogen exposure. Okay? It's going to be slower than the innate response because it's not such a generalized response. It's going to take your body a little bit of time to figure out exactly what it needs to do. And it is going to require support from the innate system. Okay, it will need that extra support in the form of things like helper T cells, okay, to help it function at its best. And it's got two parts, humoral immunity, which deals with the bodily fluids, and then cell-mediated immunity. So we've got our humoral immunity here with adaptive and our cell-mediated. Again, humoral immunity is dealing with body fluids, and cell-mediated would be specific cells. Okay, so let's look at these specific cells first, the lymphocytes that are going to be part of this adaptive immunity. Okay, so we have two primary kinds of lymphocytes here, okay? And so lymphocytes are going, Okay, so lymphocytes are going to be specialized cells. And like I said, we have two main types here. We've got B cells and we have T cells. Okay, and so our B cells are going to be made in the red bone marrow. So again, they're made in the red bone marrow. That's an M. Let's try again. So they're made in the red bone marrow. They mature in the red bone marrow. They have lots of rough endoplasmic reticulum in there, okay, because they make antibodies. Okay, and so they need lots of rough endoplasmic reticulum to help make the antibodies. And I've got three basic types of these. Okay, I have what's called naive B cells, these are B cells that have not been exposed to a pathogen yet. So basically they're inactive B cells. They have not been exposed to any kind of pathogen. And then I have what are called plasma B cells. Okay, and so a plasma B cell is activated, it's working, it's making lots of antibodies. Okay, so naive ones are not doing anything, they're inactive, they haven't been exposed to an antigen. Plasma B cells, they're activated, they've been exposed to an antigen. Okay, an antigen is our foreign particles. Okay, I cannot spell today. So an antigen is a foreign particle that's going to trigger the response. Okay, so the B cells, plasma B cells, have been activated. They're making lots of antibodies in response to an antigen. And then you also have what are called memory cells. And the memory B cells are long-lasting. Okay, they have a good memory. They're easily activated during a second exposure. These are the ones that remember the antigen, so that when they see it again, they can activate things very quickly. Okay. In T cells are going to be synthesized in the bone marrow, but they're going to mature in the thymus. Okay, so T cells are made in the bone marrow, but they mature here in the thymus. Okay, and so they have lots of ribosomes in them, lots of free ribosomes in them, so they're also making lots of proteins. Okay, and we've got 4 different types of T cells. Okay, again, I have naive cells. So these naive cells have not been exposed to antigens, so they're inactive, they haven't been exposed to antigens. Okay, I have what are called helper T cells. Okay, so helper T cells, they help B cells actually. Okay, helper T cells activate B cells. Okay, so I've got naive cells that haven't been exposed to anything, helper T cells that actually activate B cells, and then I have cytotoxic T cells. Okay, if you break that down there, you've got your cyto, which is cell, okay, and toxic, which would be poison. So these cytotoxic T cells, they destroy your infected body cells. So cytotoxic T cells do not destroy the antigen, they destroy your cell that's already sick. So they destroy an infected body cell. And then also for T cells, I've got memory cells. Again, cells that will be long-lasting, easily activated during that second exposure because they've seen it before. Okay, so let's look a little bit closer at the receptor sites on both these T cells and the B cells. So we're going to look first at a T cell receptor site. Okay, and so the T cell receptor site, this is going to attach to the antigen. Okay, so remember the antigen is the foreign particle. So in the antigen, it could be, usually it's going to be a piece of a invading cell. Okay, so the T cell receptor site is going to bind to the presented antigen, and the T cell receptor is going to be specific. Okay, so it is specific and can only bind to one type of antigen. Okay, it is specific to the antigen as well as the body protein, and we'll talk a little bit more about the body protein in just a minute. They're called MHCs. And so when a T cell is looking to bind with a foreign particle, it has to bind both with the antigen and the body protein that presents it. If both of those matches aren't made, the T cell will not be activated. So the receptor itself is made up of two polypeptide chains. Okay, I've got an alpha chain here and a beta chain here. Okay, so again, these are polypeptide chains. So like little mini proteins, right? Lots of amino acids here. So the alpha and the beta chain both have these two regions. They have a constant region and they have a variable region. Okay, so the constant region is going to be anchored in here into the cell membrane. So the constant region anchors into the cell membrane, while the variable region, that's what's going to be what actually is interacting with the pathogen. And you can see it's up here on the top. It's where the binding site is. So that's what's actually interacting with the pathogen. So that variable portion, that is what is different from T cell to T cell. So that antigen binding site on that variable region being different is what allows a variety of T cells to be able to attack a variety of pathogens. Okay? Each T cell will have over 100,000 identical T cells receptors on it. And so they can bind with a lot of antigens. Okay? And like I said, your neighboring T cells are going to be different due to different receptors up here on this antigen binding site on the variable portion of the receptor. Okay, so let's look at the B cell receptor then. So the B cell receptor is a little bit different. Remember, B cells are the ones that are going to make antibodies. T cells, we had helper T cells that activated B cells, naive T cells that are inactive, cytotoxic T cells that kill infected cells, and memory T cells. With B cells, we have those naive ones, the memory cells, and then as well we have the plasma cells that are making antibodies. So the B cell receptor is the same as an antibody. Okay, so the receptor on the B cell has the same structure as an antibody, because those B cells are going to make lots and lots of antibodies. Okay, and so those antibodies need to look like the receptor. I cannot write today. Okay, and a lot of times now you'll see antibody as AB. In my rush to make you not watch the longest video ever, I'm just making it longer by not being able to write. Okay, so your receptor of your B cell is going to have your same structure as your antibody or your AB. And so the B cell receptor, you can see it looks a little bit different than the T cell receptor. Okay, so the B cell receptor can attach to two antigens. If you remember on that T cell receptor, it just went straight up and just had one antigen binding site. B cell receptors have two antigen binding sites. Okay, so they're made up of 4 polypeptide chains. Okay, so I've got my 4 polypeptide chains here, and they're held together by a disulfide bridge. Remember, a disulfide bridge, that's a pretty strong bond. Okay, it's a covalent bond between 2 sulfurs, so it's a pretty strong bond. Okay, so they've got identical heavy chains and identical light chains. Okay, so the heavy chain here is identical, and then they've got identical light chains, but again, they have that variable region. And so the variable region is going to change. Okay, and you can see how that's, again, our antigen binding site. So that's where we're going to get the variety of which antigens they're able to recognize. So like I said, the antibody is going to have the same receptor. Similar to the T cell, you can have 100,000 receptors on each of these B cells. And again, they'll be different from their neighboring B cell because they will have different receptors. Different receptors will make each cell different, just like we've been talking about. Let's take just a second to just kind of look at the two, do a little compare, contrast here between a B cell receptor versus a T cell receptor. So again, they both have that antigen binding site, okay, but remember the T cell receptor just has one, B cell receptor has two, okay. They're both going to have variable and constant regions. So they're both going to have variable and constant regions, okay. They're both going to be made up of polypeptide chains. Just remember your B cells have 4, T cells have 2 polypeptide chains. Okay, and then they'll also, like I said, they'll be different from T cell to T cell and B cell to B cell. Okay, but you'll have lots of each one of these receptors on both B cells and T cells. Okay, so let's take these cells and kind of go a step up here and apply it to tissues. So we've got primary lymphatic tissue here, or primary immune tissue, which is going to be things like our bone marrow here, okay, where those B cells and the T cells are made, okay, as well as the thymus, where they're maturing, where those T cells mature. So that's our primary lymphatic tissue here, okay, the thymus and the bone marrow. The secondary lymphatic tissue is where the cells that are mature, so these guys right here, these B cells and these T cells that are mature, this is where they will go wait to be activated, okay, and these are things like the adenoids, which are actually inflamed tonsils, okay, and our tonsils in the back of our throat, okay, lymph nodes. We've got lymph nodes, a high concentration of lymph nodes in the neck, high concentration of lymph nodes down in the groin, in the armpit region. Okay, those are the highest concentration of lymph nodes in those 3 areas. Okay, the spleen, Peyer's patches, which are little patches of immune tissue found in the small intestine, almost like tonsils for the small intestine. Okay, the appendix is thought to have some immune function. Okay, so all of these things are different forms of secondary immune tissue. Again, where the mature cells go to wait. So they go here, they wait to be activated, and then when they're activated, they'll travel about and do what they need to do. Okay, so just a quick kind of refresher, if you don't remember. Okay, so let's talk for a second about lymph tissue, or lymph fluid. Okay, so lymph fluid is the fluid that is going to end up in the lymphatic capillaries, or the lymphatic vessels. It is basically has a very similar composition to the interstitial fluid, similar to blood, but it doesn't have the blood cells. Okay, lymph fluid will end up back into the blood. Okay, it will circulate through the lymphatic capillaries, usually get filtered through the lymph nodes. It's one of the ways we can help filter our body system. Like I said, all of the lymph fluid comes from, like I said, blood that was not picked up into the capillaries. And so it's going to end up back in blood circulation. And so it is going to, it's just going to help filter some of the fluids in our body. Okay, in addition to the lymph fluid, okay, if you hear lymphatic vessels or lymphatic capillaries, okay, lymphatic veins. Okay, so the lymphatic vessels, they act like veins. They're going to be they move one direction. They're taking the fluid. They're actually going to take it, like I said, back towards the heart, because you can see here, you've got these lymphatic ducts that are entering in with the veins. That's where the lymphatic fluid will drain once it's gone through the lymph nodes. And so the lymph nodes themselves, you can see, here's a lymph node over here. Okay, and the lymph node themselves, they have plenty of lymphocytes and macrophages in them. And the lymph fluid will come out. It will go in and it will get filtered through those lymphocytes and macrophages, reticular tissue in there that acts almost like a screen